In today's lesson, we will study about the genetic engineering and its implication. At the end of the video, you are expected to achieve the following learning targets. Humans have selectively bred plants and animals for thousands of years. Selective breeding is a form of artificial selection whereby man intervenes in the breeding of species to produce desired traits in offspring. In here, breeders select parents with desirable traits and cross them together. From their offspring, they again choose the best combination of traits, and the process continues over many generations. By breeding members of a species with the desired trait, the trait's frequency becomes more common in successive generations. Selective breeding of plant crops has allowed for the generation of new types of foods from the same ancestral plant source. Here are some examples of crops before humans have started cultivating them. We have here banana, corn, tomato, eggplant, and strawberry. On the other hand, selective breeding of domesticated animals has also resulted in the generation of diverse breeds of offspring. Horses, for example, have been selectively bred to become either as race horses for their speed or as draft horses for power and endurance. Belgian blue cows have been bred to produce more edible lean meat, and dogs are selectively bred for particular traits such as for hunting, herding, racing, or as toy dogs. Selective breeding is under an umbrella term called classical breeding. Classical breeding makes use of existing naturally present gene variants in a species and makes use of that natural processes of sexual and asexual reproduction in breeding. But there is another way by which an organism's genetic characteristics can be changed, and that is through genetic engineering. Genetic engineering involves a direct change to an organism's genome in the laboratory. Traits may be modified by introducing new traits into an organism, enhancing present traits by increasing the expression of the desirable gene, or disrupting the expression of some undesirable genes. The term biotechnology usually means the manipulation of genes for some practical purpose. And for this lesson, we will identify some ways by which genes are engineered. First, what is recombinant DNA technology? A recombinant DNA contains genes from two or more different sources. This one here is a simplified diagram of gene cloning using a recombinant DNA. Pieces of DNA such as human DNA can be engineered in a fashion that allows them to be copied or replicated in bacteria or yeast. To make a recombinant DNA, a researcher needs a vector. This is a piece of DNA that acts as a carrier for the foreign DNA. One common vector is a plasmid, which is a small accessory ring of DNA found in bacterial cells. The sample bacteria in here is an Escherichia coli or E. coli. Next, to introduce the foreign DNA, in this example, the insulin gene from a human cell, two enzymes of the plasmid are needed. First, the restriction enzymes will cleave or cut the DNA at specific places. For example, restriction enzyme EcoR1 always cuts the DNA between the base sequences G, A, T, T, and C. With the gap in the plasmid, a piece of the human insulin gene will be cut in the bases complementary to those exposed by the restriction enzyme. Next, the DNA ligase will seal the foreign DNA into the cut plasmid. Molecular biologists treat the bacterial cell to make them more permeable and place back the now recombinant plasmid. As the bacteria replicate the plasmid, the gene is cloned. The production of multiple copies of a single gene is a type of DNA cloning called gene cloning. From here, a scientist may allow the genetically modified bacterial cells to express the cloned gene and retrieve the protein. Or they can remove copies of the cloned gene from the bacterial cells and then introduce it into another organism, such as a corn plant, to produce a transgenic organism. Now that you learned the key steps in recombinant DNA technology, let us describe the ways in which these plasmids may be introduced back into host organisms. Biolistic is short for biological ballistic. It is the method of directly shooting DNA fragments into plant cells. Micropellets of gold or tungsten is coated with these DNA fragments and are released from a gene gun through a high pressure helium gas to directly penetrate the cell wall of the host. Gold and tungsten are used because they are not chemically reactive. Cells now that survive the bombardment will acquire the ability to express the designed protein. Another example is the heat shock treatment. In this method, both the plasmid DNA and the bacterial cell is incubated together initially at 4 degrees Celsius for 30 minutes. They are placed in a calcium-rich solution to counteract the electrostatic repulsion between them. Next, the temperature of the environment is suddenly increased into 42 degrees Celsius for a minute, causing now the porous sizes of its cell membrane to open, allowing the entry of plasmid DNA. Afterwards, the temperature is decreased to 4 degrees Celsius for 2 minutes. The cells that took up the plasmids acquire new traits and are said to be transformed. Meanwhile, as an alternate approach to chemical transformation, an electrical field can be applied to the cells to enhance the uptake of DNA. This method is known as electroporation. This technique has been used to introduce nucleotides, DNA, RNA, proteins, carbohydrates, dyes, and virus particles into prokaryotic and eukaryotic cells. Now, how can we check the presence of the recombinant DNA plasmids after they have been copied many times in host cells? One technique used is gel electrophoresis. It is a method of separating DNA fragments according to their size. During this process, DNA samples are loaded into wells at one end of a gel that is negatively charged, and an electric current is applied to pull them to the opposite side that is positively charged. Due to molecular weight, DNA fragments that are smaller moved farther through the gel than larger fragments. This now results in a pattern of distinctive bands called a DNA profile or DNA fingerprint. Each bands represent a group of same-sized DNA fragments. But what is this technique commonly used for? Using DNA fingerprinting, genetic differences between individuals can be detected. One good use of this method is for identifying the parents of a child or identify the remains of someone who has died, such as a victim of a natural disaster. Some key applications of this technique are also listed in here. Another method for screening the presence of the desired gene in the inserted plasmids may be confirmed using the PCR. PCR is short for polymerase chain reaction, and it is a technique used to amplify even few pieces of DNA in the lab. PCR involves three basic steps. First, the DNA is denatured for the two strands to separate. Then, these strands are given nucleotide primers which will begin adding bases in the strands later on. And lastly, extension, where a DNA polymerase will add complementary bases to each of the single DNA strand. In this diagram, you will notice that for every cycle of the PCR process, the number of DNA segments double in number. But why do we need to make multiple copies of the DNA? DNA amplified by PCR is often analyzed for various purposes. Because so little DNA is required for PCR to be effective, it is commonly used as a forensic method for analyzing DNA found at crime scenes, with only a drop of semen, a flake of skin, or even the root of a single hair. Let us now identify organisms whose DNA has been altered using genetic engineering techniques. One example of GMO is bacteria. Some naturally occurring bacteria that eat oil have been genetically modified to do this even more efficiently and are used in cleaning up beaches after oil spills. These bacteria are given suicide genes later on, which will cause for them to self-destruct when their job has been accomplished. Next, we have our genetically modified plants. For example, the flavor saver tomato. It was the first genetically modified organism that was licensed for human consumption. The trait modified in this tomato is its ripening process. Inhibiting the undesirable gene delayed the softening of the tomato and extended its shelf life. Another example is the BT corn. BT corn is enhanced through biotechnology to protect against insect pests. Its built-in insect protection comes from a bacteria called Bacillus thuringiensis or BT. The protein produced by BT corn selectively targets caterpillars that feed on these plants. This same technology has been also applied in the Philippines for the development of BT eggplants. Like plants, animals can be genetically modified to increase their value as food products. For example, a new form of transgenic salmon contains genes from two other fish species, which allows it to grow quicker. The Aedes aegypti mosquito, on the other hand, acts as a vector for several human diseases. A transgenic form of the mosquito contains a genetic kill switch that kills its offsprings, thus reducing their population size. Despite the proposed benefits of GMOs, some people have raised their concerns regarding the consumption of these modified foods. Because of these issues, manufacturers are urged to provide labels that notify consumers of GMO presence in their products. After all, it is the consumer's right to make their own choices. That's it for today, class. Thank you so much for listening.